Dr. Alfred Sun came to Singapore in 1997 and studied at the Chinese high school under full scholarship from MOE. Subsequently, Alfred was awarded the A-star and SS PhD scholarship and obtained his Bachelor's of Science from Duke University and PhD from Stanford University. During graduate school, Alfred pioneered a novel transdifferentiation method to convert human dermal cells into functional neurons that ignited his interest in human cell-based modeling of neural development and degeneration. Currently, Alfred is focusing on functional studies of neuronal subtypes and also exploring 3D brain organoid models as a novel tool to study neurodevelopmental disorders as well as aging-linked neurodegeneration. So here you go. We have Dr. Alfred Sun. So please go ahead and do a round mic check. We should good to go. Thank you, Dr. Alfred. The floor is yours now.
Thank you, Callum, for the very kind introduction. Can everybody hear me?
Yes.
Great. Okay. So, the title of my talk today is actually investigating human neurological disorders with human neural cells. As being introduced, I'm primarily interested in understanding human neurological disease, and the reason is really notwithstanding the kind of knowledge and insight we learned from model organisms shown here. Those are commonly used laboratory organisms for studies. I just want to remind everybody that there's a vast difference between human system versus all the animals we use in the lab, right? So that really comes from the next slide that summarizes 3 of the major distinctions among the human versus the other animal species. For example, our brains are really big and complex, as shown here, as compared to the mouse or the rat brains, which is a lot smaller and less complicated. Also, so not only the size, but also the structures and the unique features of the human brain that made us unique. So I'm illustrating here two of such differences. One is in the developmental sort of developing brain. You have structures known as the OSVZ zones that's uniquely present in the primate and above. It's virtually absent in the mouse or rat brains. And also in the case, this is a mid-section of the midbrain part. This is known as the substantia nigra region. Now this is, I will come back to this later in the second half of my talk, but here you can clearly see this is pigmented, right? These are dark regions. So this sort of pigmented is due to what we call neuromelanin. The presence of neuromelanin is also primate specific and it's age dependent. So it's in you and I, and probably a lot darker in the elderly brain, but it's not in the mice, even in the aged mice or rats. So those are the features that are uniquely present in the humans. Some of the unique features that are uniquely present in the human, but not in the other species. And of course, one last reason is our brain is really inaccessible. So it's really well protected from the scalp. So anything you want to do to it, it's pretty much impossible. So having said all this, is really to pave way to suggest that we really need some kind of models, human neural models, to have that in a dish, to study them and be able to model certain phenotypes or pathology in a dish. So let's take a sort of quick overview of the current status of the field when it comes to human neural cells. How do we obtain those things? So the first arrow here in black is shown as the iPS cells. This is nicely shown by Xuyang and Yamanaka. So from a somatic cell, we can be blood or skin cells, we can convert them into ES cells or iPS cells. So for our purposes, these cells, we put these cells as functionally equivalent. And that you can do also what we call classical differentiation using different patterning factors and all that stuff to make them into new progenitor cells and then into another round of maturation into neurons. Now this is fine, but this is what scientists have been doing for the last 20 decades or 30 decades, and we learn a lot, okay? But there are certain caveats with this system. That is, it takes longer time, and usually you get a mixed neuron types and variable efficiencies. So I was in grad school, so we thought about this way as a direct conversion. So starting from dermal fibroblasts, we put in genetic materials, transcription factors, or microRNAs, and then that can directly convert into neurons. So this is what we call a direct conversion or transdifferentiation by many other people. It's cool, but it cut down the time required a lot, but it has also its downside. For example, the yield is typically lower, which means you can't do a screen, and there's limited subtypes, and the cells are typically immature. So when I started my postdoc, I decided to study this diagram quite a bit and decided to come out another arrow that I want to draw onto the figure schematic here. So what about starting from ES cells? Can you directly take them to neurons? The advantage of starting from ES cells is that these cells are proliferating, and you essentially have a line. So you don't have a problem with the initial cell number. You don't have a problem with sort of the line-to-line variation and the passage variation this way. So if you can figure out this, then it will also be quite a fast way to get functional neurons. So that's exactly what I getting focused on in my first part of my postdoc. And that work has been essentially published in 2016. So essentially I come up with two genetic material sets that upon overexpression in pluripotent stem cells, ES cells, iPS cells, you will take them either to excitatory neurons or inhibitory neurons, the two principal neurons of our cortex. So we call that IEN, stands for induced excitatory neuron, and IGN, induced for induced inhibitory neurons. Okay. So just to give you a quick overview about how robust this system is and how fast the churning of the neuron is, you can see that within days, this is probably within a week, you will see the cells really just burst out from their compact ESL-like morphology to take on the shape of that of neuron. And by day 20, you have a really elaboration of the neurites, and the cells are very mature. So it is armed with this sort of novel direct conversion method. We realize that now we can really utilize such protocols to really get some functional human neuron and be able to study if there are any functional phenotypes. So that leads to the second story, a quick one I want to talk about, that is Angelman syndrome, right? So Angelman syndrome is a very rare childhood neurodevelopmental disorder. We choose that actually primarily because it's monogenetic. So we know the genetics very well. That is the UBE3A deficiency. So you can see here, in the majority of the cases, if you have a deletion in the maternal copy of this UBE3 because it's imprinted, you have a problem. And then in other cases, you actually have mutations or imprinting defects. All this suggests that if you cause some changes to essentially loss of function mutations or loss of function phenotype of UBE3A will give rise to Angelman syndrome. Okay, now Angelman syndrome as a syndrome has different spectrum of phenotypes, right? But the one that we really wanted to focus on is actually epilepsy, seizures, as shown here. Because why? Because actually this one is the leading cause of death for those patients. When I was in the States, a pediatrician told me that for Angelman cases, if the child is able to survive into adulthood, they're relatively okay. They're a little bit impaired, they're intellectually impaired, but by and large, they lead a normal life, all right? However, many of them die at a young age, at the teenager stage. And the reason is really pathetic. That is, many of them die in drowning. And why? It's because sometimes the spontaneous erratic seizure happen when they are swimming and they just get drowned. So that's really pitiful. And so we want to understand how seizure happens, or why the Angelman syndrome patients have such a high propensity towards seizures. Okay, so here is the sort of the concept and the study design. We want to understand how UBE3 loss of function will eventually lead to epilepsy in patients. And here is the study design. So we use CRISPR to knock it out UBE3 in the embryonic stem cells. We use H9, a well-known used line in the world, and also we have some Angelman patient-derived iPS cell that were obtained commercially. We then make them into induced neurons and then study the functional deficits. And by doing that, we hope to gain some mechanistic understanding about the disease and how the seizure will be ensued. Okay, so here is the data. So because this story has been published, so allow me to go through this really quickly. So here you can see that we use CRISPR-Cas9 to knock it out, and we validated that by both Western blotting shown here. This is Azure, and also to use immunostaining. So in the neurons, in the wild-type cells, you have very nice UBE3 staining, but it's completely absent in the knockout. So this is just validated that we have that. Well, when we make neurons, we realize actually the neurons we made, actually they just behave they just become neuron just as fine as the wild type cells. And actually, when we look at it, we cannot tell any differences in terms of morphology. When we look at the show analysis, the neurons interaction, they are normal. Synaptic density, excuse me, is also normal as quantified here. This prompts us, this sort of lack of apparent morphological phenotypes. It's kind of surprising and not surprising. So it is surprising to us that this looks absolutely fine, and it is, and yet in the field that many people have reported all kinds of phenotypes with this dendrite phenotype or whatever in the mouse models. There's a bunch of other phenotypes, but we don't see that in the human neurons that were obtained. But it's probably not surprising to us because Angelman syndrome, by and large, during development, you have this seizure ability, but their development is by and large normal. So this implies to us in this UBH3A knockout that we probably don't expect to see a major sort of differentiation phenotype. Now, but this also then allowed us, sort of propelled us to look into the function of the neuron more carefully. Actually now, and then we see that actually at the higher current injections, these knockout neurons are actually able now firing a lot more. So the spike frequency is increased in the knockout neurons as compared to the wild type, which is quantified here. To be sure, this is not a line specific thing because I mentioned that the first clone was generated, the first knockout clone was generated in H9. We just want to be sure that we can do that in another pair, right? So we choose H1, and this is what we call clone B. So in both cases, fortunately, we are able to reproduce the phenotype. So what is the phenotype? This is actually called a fast AHP, which stands for after hyperpolarization. So we realize that actually in the knockout cells, it has an increased amplitude of the FAHP. And we show that in both the H9-derived clone, knockout clone, as well as in the H1-derived knockout clone. Okay? And if UBE3 is really mediating this effect, then one would predict that the exogenous restore of the UBE3 expression should rescue the phenotype, right? And that's what we did, and this is exactly what we saw. So we see that lentiviral-mediated overexpression of UBE3A was able to restore the spike frequency phenotype as back to normal, and also decreased the FHP amplitude to the wild-type level. All right. So I mentioned all this so far. The two clones are generated using the human ES cells. What about in iPS cells, right? We had the iPS cell, we made it into neurons, but here it's quite the opposite, right? So the Angelman cells, as expected, will not have UBE3A because they have an imprinting problem. Now, if we put back UBE3A, it should express it, and that's what we see. We indeed express it, but the phenotype is expected. It's the opposite. You would expect this to behave like a wild type, this to behave like a knockout, and that's what we see here. Okay, so far the data is quite nice, that it's all very consistent. But we want to understand a little bit more about the network level, because eventually it is a network hyperactivity, a seizure-like activity. But what the phenotype I told you so far is only a cell intrinsic sort of deficit in terms of finding, right? So to do that, we turn into another assay known as a multi-electrode array, also known as NEA. So we essentially play neurons onto the gold-plated electrodes and measure the electrical activities of this ensemble of neurons, the hundreds and hundreds of these neurons. And we'll be able to document their sort of burst firing like this. And indeed, upon quantification, we realize in the knockout that there is a higher synchrony or higher frequency of burst-like firing in the neurons for the knockout. Okay, this is perhaps better visualized through a movie here. Okay, so you can see that the knockout cells seem to have this synchronized bursting a lot more often than the wild-type cells. All right, so this is what I showed you so far. So I showed you that UB3 loss cause FHP increase, excitability also increased, and that leads to a network dysfunction. But what's mediating this, right? So that's where we look at what we search the literature essentially to look for what are the potential molecular players underlying this increased FHP, and there's not much players known actually. So the most highly likely player is actually the BK channel, and BK channel is well known because they are very good pharmacology, use pharmacological agonist and antagonist for it. So we try that, and then we use essentially Paxilid, which is a well-known validated BK blocker. So indeed, we show first that the BK current in the knockout cells are larger than in the wild-type cells. And so to understand this better, why the BK current is actually higher in the knockout than the wild type, we turn to quantitative Western blotting. And this is where we got a lot of help actually, but from the Azure C600 imaging system. So it's shown here. So we notice that in this triplicate pairs, that the only thing really consistently change is the BK. But you notice the level of changes is not that high. It's only about 20%. So we can see this because BK never gets changed a lot. It's almost impossible to change the BK level a lot. So it's about 20% change, but this 20% change is actually quite meaningful. It's causing a phenotype. So I have to say that in this particular case, in this Western, the BK and a lot, for example, the ATP, the ATPase alpha subunit will actually image from the same membrane thanks to the NNR near-infrared imaging system from the Azure system allows me to be really sure that there's no stripping that's involved, to be very sure about the quantitative changes in the protein levels of those things. All right. So an alternative way to confirm that there's a BK increase, we turn to atomic force microscopy. This works slightly differently. Essentially, it's a cantilever that allows you to functionalize by putting a BK antibody to it. Now, if you scan this thing across the neuron cultures, if there is a BK, right? So the BK will attract the antibody, will bind to it, and that is shown as a registered S3. It's shown as a downward force dip, and that's registered by this cantilever, and it's shown here. So by doing that and plotting the distribution of the force, we can actually have an idea about the BK density. So by doing that, that's what we show here. So it looks like the neuronal surface have a higher BK density in the knockout as compared to the wild type. So putting these two independent evidence together, we conclude that indeed there seems to be an increasing functional BK surface from the knockout neurons as compared to the wild type. All right. So having shown that, then the next thing we want to show is actually the BK blocker now can normalize the function, right? It's a phenotype. Yes, indeed. So we show that when you apply the Paxilin at the appropriate dose, you're able to rescue all this phenotype I told you about, the excitability, the FAHP, as well as the network level. Its hyperactivity is restored to the normal level. Okay, so I'm just, in the interest of time, I'm going to skip all this Western to the one panel of Western data. Essentially, this panel work is to show, to nail it down, to show that the UB3 actually function as the E3 ubiquitin ligase to degrade. So it's through the proteomimetic degradation pathway of the BK. Okay, again, all these images were done using the Azure machine. So that I can get to the more meaningful stuff later. Okay, so I just showed you that we find that the substrate of a novel substrate of UB3 in this case is actually the BK channel. Now this is a very novel finding. Nobody in the field of epilepsy or I would say Angelman Syndrome ever suspected that this is a player. So this is a novel finding, and it's a novel substrate for UB3. And we also find that this can be used as a way to intervene with potentially to normalize the phenotype, right? But there's one thing missing that I didn't get to show you. Oh, that is from, again, I jump ahead of myself. But the data so far I showed you is all from the 2D cells, right? And it's not a normal differentiation protocol that I use. We use the direct conversion, which is fast and functional. But there was a question, there was a concern from us, from the reviewers, that how is this, if you do it in a normal developmental differentiation protocol, would this phenotype still be there? So we thought about that, and the way to address that, we decided to use the 3D organoid approach. So we make a normal cortical brain organoids. From this, you can see that at day 30, both the wild type and knockout give rise to very similar organoids. And if you session them, you realize and look for the protein markers for the cortical organoid, they are by and large normal as well. There's no discernible differences between the wild type and knockout, and as far as we can tell. Okay, you grow them longer to 100 days and above. Now this really grow to a millimeter scale organoid, and if you section them, you can see cortical layers. I have to admit that based in our hands, this is a modification from the Pascal protocol that we used, Pascal's protocol we used. We can't really get this very nice 6-layer cortical layer, layer by layer like that. What we have is a rough segregation of the upper and the lower and the deeper, upper and deep layers, okay, as shown here. But again, between wild type and knockout, there's no major difference in terms of the layer marker distribution. That's my point. So again, we see that that actually BK is increased in the knockout, and all the phenotypes that I mentioned about that we discovered through the 2D neurons are actually can be recapitulated as also present in the 3D organized. Okay, but having 3D organized actually is advantage, allowing us to look at the network analysis more carefully using calcium imaging. So this is what we did here. So using 2-photon calcium imaging, we're able to show that the what we call a synchrony index, which is the network level simultaneous firing of different neurons, is actually increased in the knockout as compared to the wild type. Again, highlighting the hyperactive network activity of the organoids. Okay, so this is what I showed you so far, leading to a network activity. But really, the patient has epilepsy, right? How do we have epilepsy in a dish? That's a fundamental question that we cannot address using all the methods I told you about. To that, we can only go back to the animals. So we obtained an Angelman mouse model from University of North Carolina in the States, and then we shipped it over and performed we decided to try to see if first validated that the Angelman mouse, the knockout mice, are actually prone to epilepsy. And the second, to show that indeed we patched that we realized, hey, in the hippocampal neurons in this case, that there is an increase in the BK current and the AHP is also increased. It's the excitability of the neurons also increased. This is again the novel finding. None of the Angelman mouse paper before mentioned this phenotype. Okay, but really the killer experiment is this one, that we have to show that now the BK blocker, Hexlin or and other stuff, should be able to rescue the epilepsy phenotype. So we use two paradigms, looking at epilepsy threshold using chlorophyll, as well as epilepsy grade using picotoxin. This is a GABA blocker. In both cases, we are able to see that the Hexlin is able to normalize the epilepsy propensity of the mice. All right, so to quickly summarize, this is published end of last year. So we started actually, this is really a kind of a functional modeling part of Angelman patient Angelman syndrome neuron cells. We started from a very simple idea, that is, we want to make a functional 2D neurons and study whatever is wrong in this neuron. And surprisingly, this led us to uncover excitability phenotype of the knockout neurons. And we subsequently validated these findings in 3D human brain organoids. And we did a bit of biochemistry to find out how it works. And then we went on to the mice and be able to show that indeed that the epilepsy susceptibility is able to be rescued using the BK blocker. So that is sort of quite a complete story, and I think that's probably one of the reasons why it gets into science. Okay, so I just want to highlight a point from here that's different from most of the Angelman studies in the field, that most people study the mice neurons and mice models. We learn a lot from these mice models, but I also mentioned the mice studies. None of these mice studies uncover this phenotype, right? So why is that so? And what's so special about our system? Well, I would think that there are at least two things. One, there could be a mice-human difference in the sense that the mice phenotype is there, but the phenotype, the magnitude of the phenotype or the effect size of the phenotype is a bit smaller in the mice as compared to the human. For some reason, for humans, the AHP change is really apparent. We're able to see that very early on from the study. Okay, in the mice, it's not so apparent. So perhaps that escapes the investigator's attention. But really, what we are doing differently is we use this transdifferentiation or direct conversion protocol that our neurons are really functional. Okay, that's very different from the traditional protocol where you have to culture the neuron for a long term, and still many of the neurons do not fire, or some of the neurons fire a lot. It's electrically more active than the others. We don't have that. So in this case, the functional maturity of the neurons really helped us to uncover this novel phenotype. That's what I believe. Okay, then just to the worst thing that can happen to a postdoc at that time when you're about to really halfway through your work is your boss left. So I had to shift to my second postdoc, and then the inevitable thing is that I have to shift the project, right? And so the new PI that I worked with is actually a clinician focusing on Parkinson's. So he said it's fine, you can work under me, but you have to switch to Parkinson's modeling. So that lens actually forced me to look into the organoid model a little bit more, and was able and I'm about to share the kind of to share some work coming from this study, because now actually surprisingly this is becoming the major part of my team's focus. So to understand Parkinson's, which is neurodegeneration phenotype, we first know that there's a very particular notable feature of the pathology of Parkinson's. It is a very special type of neurons, dopamine neurons, and it is only the dopamine neurons in the substantia nigra, which is a region within the midbrain that is dying. Okay, so the pathology is really clear. So we thought that time that in order to understand that, perhaps we can make a human midbrain organoid system. That time we are really inspired by Lancaster's work about the whole brain organoid. But we think that for the Parkinson's, a midbrain specific organoid may be more useful. So again, this is past work. This has been published in Cell Stem Cell in 2016. So just to give you, this is again, we learned from developmental biology to know how to pattern the ES cells into the midbrain region by sort of optimization of the two axes, the dorsal ventral axis, as well as the rostral-caudal axis using morphogens, solid hedgehog, and then Wnt pathway. Okay, this is to show how the organoid will grow. So essentially, they grow to more than 2 millimeter big over the course of 50 days. And we do immunostaining to look at the organoids. We show that they are really authentic midbrain organoids that behave as far as we can tell, as compared to the mouse ventral midbrain sessions. You can see that they are marked by the cardinal midbrain of sort of floor plate markers. These are FOXA2 and OTF2. And later on, these cells later become differentiated neurons and turn on TH. And we have subtype of those TH neurons as well, marked by PERK2 and binding. And these cells are functional through our electrophysiological analysis. Okay. And we did a RNA-seq analysis, look at the global transcriptome of the cells, and compare to adult midbrain, adult fetal, adult forebrain, or adult other brain region. We realized that our we call that midbrain-like organoid. So our organoids actually lies closer to the perinatal midbrain than any other brain regions, or than the 2D dopamine neuron that people typically produce. Okay, so that's encouraging. And we also demonstrated that through single-cell RNA, this is Fluidigm technology at that time, that we are able to have the presence of several types of cells which present in the ventral midbrain. For example, in terms of the inhibitory neurons marked by GABA. So the question is now, you can make this within a month, but what happens if you grow them in the long term, right? 3 months later. So we saw this. So there are black spots coming out of this. These are not contamination as what we initially thought they are. Actually, they are very encouraging signs because these are the neuromelanins that I mentioned earlier in my talk. So these are the neuromelanins present specifically in the substantial region of the midbrain. So this is a very important finding because I told you that none of the mouse models produce it. In fact, the mice don't have neuromelanin, right? But yet, surprisingly, and none of the earlier works produced in vitro sort of spontaneous formation of neuromelanin. This is the first case that a human in vitro neural cell aggregate is able, or cell type is able to show this neuromelanin. Of course, we have to validate a bit more before claiming this neuromelanin. So we look into the structures and look at the AIE stainings compared to a post-mortem human adult midbrain regions to see these are really the black pigments that look similar to ours. And then we also look at the structures of these neuromelanin granules isolated from organoids and compared to the neuromelanin granules isolated from the post-mortem human brain tissue. And again, they look very similar. Now, this is a proof that I showed you. So the human ones are black, but not the mouse counterpart. So we take mouse ES cells, make it undergoing the same protocol to make a mouse midbrain organoid, which contain TH neurons, okay? But they're not black, okay? So this leads me to almost the end of the talk. So currently, what I'm really trying to do is to use sort of kind of innovative human neural stem cell technology to really understand human neurodevelopment and pathogenesis. And a major focus of my current team is actually neurodegeneration in Parkinson's disease, utilizing these human midbrain organoids. Of course, I mentioned development because I'm also very interested in pediatric abnormal neurodevelopmental diseases, much like the Angelman's case. Okay, then a little bit, but to essentially this is what I'm trying to do, right? You have a dish of organoids, and we want to use this to model a really age-linked neurodegeneration disease such as Parkinson's, which has certain pathological hallmarks, right? One is a Th neuron degeneration, as you can see by the depigmentation in the substance nigra in the patient's brain, as well as abnormal protein aggregation. This particular is known as Lewy body. All right. So the question is, we are very active in pursuing this direction to show that how can our midbrain organoid be able to recapitulate those disease phenotype in the dish. And if we can do that, then how can we leverage this knowledge to understand or to support development of the Parkinson's disease? All right. As I mentioned, so I'm currently a junior PI, but actually I will be moving, and I will be joining Duke and US Medical School as beginning early next year as an assistant professor. So my brand new lab will be seeking technician students and postdocs. And if you have any questions, my email is here. Okay. And the last slide is really perhaps the most important part of this acknowledgement to those other institutes that I've worked with, National Neuroscience Institute of Singapore, the Genome Institute of Singapore, as well as Duke and US Medical School. And of course, this talk is not possible if Azure didn't ask me. So of course we have to give credit, but this is fresh that I take this early this morning. Okay, so this is the machine that we're using right in my lab, and we use it on a daily basis. So the one advantage I really like about this machine is it has a very small size, so it's a very compact machine that it can use for various purposes. Okay, I guess I will stop here, and then if you guys have take any questions you guys have.
Thank you, Dr. Alfred Sun, for the awesome presentation. This webinar is hosted by Azure Systems. We aim to build products that bring real and substantial value to scientists. So to accomplish this vision, we simplify the way scientists work by innovating in unexpected places. Equipment-wise, we are currently having, let me put on the pointer. Okay, Sapphire Biomolecular Imager, the first hybrid scanning system with ultimate scanning speed and flexible application. And we also have Azure imaging system, a better improved version of Azure C-series imaging system that Dr. Alfred used in his research. And the third one will be Azure Cielo real-time PCR system, our newly launched reliable qPCR system. So besides that, we have a variety of reagents and Western blotting accessories, such as total protein staining for your Western blotting needs. So this is Azure imaging system, where Azure 600 is having the same specification as Azure C600 imaging system, which Dr. Alfred used to visualize protein knockout in Western blot. Okay, each Azure imaging system provides flexibility, quantitative accuracy, intelligent workflow that gives you data integrity that meets publication standards. So these are the list of applications that can be performed with Azure imaging systems. It covers protein expression imaging, a protein DNA gel, and also bioluminescence bacteria. You can choose your system based on your need, and here we have a total of 6 models for you to choose from, and they all can be upgraded. If you're interested in Azure imaging system or any of our products, please check out our app's website. Now comes with BIOS product citation. BIOS is a search engine for life science experimentation. So here you can check out who and which research are using this equipment. It's very good for referencing. Okay, so thank you for your time here with us today. A huge thank you to Dr. Alfred Sun. If you have any inquiries, please feel free to contact us via the email shown on the screen, or you can contact your local distributor and we will follow up from there. All right? So thanks again for joining this live webinar. Thank you, Dr. Alfred Sun. I'm Kailing, so the Regional Field Application Scientist for Azure Biosystem. Here today with us, we have Nicole and also the Vikram. Before you go, please fill in the webinar feedback form to help us improve and shape our future webinar according to your need. So the link will be pasted in the chat box, or you can scan this QR code with your phone and give us your feedback. Okay, thank you everyone for coming. Thank you, Dr. Alfred again.
Thanks, Dr. Alfred. Thanks, Kailing.
Thank you.
See you again. Bye.
See you again. Everyone, stay safe. Bye-bye.